Okay, so let's pick it up on the third part of prokaryotes here. We left off discussing some of the differences between bacteria and archaea and a little bit of the differences to the eukarya. Again, which is why the phylogenetic tree looks like this. Common ancestor first gives rise to bacteria, then goes down that evolutionary line, which eventually gave rise to archaea, and then eventually eukarya. So evolutionarily, throughout the history of the Earth, when we look at when did life appear, bacteria were first, archaea were second, and then the eukarya were the third group to evolve. Okay, so I want to briefly jump into the archaea, talk just a little bit about these guys because there's some really important concepts here and some relevance, but we're going to primarily focus on the bacteria. Part of it is there's just so much more known about bacteria than archaea, which leads to a huge opportunity. I encourage you guys, check this out, come back in 10 years and tell me what you've learned and that you're now the country's leading archaea expert. Would love to hear that. So, all right, archaea generally can be found in crazy extreme environments. Extremely hot, extremely salty, lacking oxygen, environments that other life just has no chance in survival. These guys can live there. That's why we're still learning about them, because it's difficult to study an organism that lives in a volcano or lives in a sulfur flat or a salt flat or the intestinal tract and things like that. It's very difficult to try to bring them into the laboratory and recreate their natural environmental conditions. In general, archaea are grouped into three categories. These are not kingdoms or phylum or anything like that, just kind of groupings for us to kind of put them into perspective. First grouping is what we call the extreme halophiles. Halo refers to salt. Phyle or philic means you love it. So these guys love salty or saline environments. That's where they thrive. So if you go to the salt flats along the coast where you're like, man, there's nothing here. There's no crabs, there's no fish, there's no birds, there's no weeds, there's no algae. Odds are you're going to find some archaea there. Or maybe nobody's looked. There's a great opportunity. Dig up a sample. Take it back to the lab. See if you can find the evidence of life in it. Odds are there's going to be something there. Second grouping are the extreme thermophiles. Thermo refers to temperature, especially hot. These guys love hot environments. This is where the volcano comes into the picture. There's actually life in the crater of a volcano. Now our challenge is, how do we go in there and get a sample of it? That's not a real safe environment. But as technology continues to grow and improve, scientists are finding ways to do this. It's crazy to think a scientist actually goes into a volcano and gets a sample to study, but people are doing it. So again, that would be just an insane job. But somebody's doing it, and our knowledge is growing. The third category are the methanogens. These guys live in very low oxygen environments, anaerobic environments often, and what they do is produce methane as a waste product. So methane, if you guys remember methane, CH4, carbon with 4 hydrogens, or let's draw out the chemical structure. That's that gas produced when carbohydrates are broken down, glucose is broken down, and it gives off methane. The hydrogens combine with the carbon, producing methane as a gas. So our intestinal tract, we have these guys in it, and they're producing the methane that gets released. Cows, pigs, horses, all sorts of mammals have methanogens living in their digestive tract that are partially responsible for methane production. All right, so some interesting organisms, some that we just don't know a lot about them. We definitely need people to go look at these and get more knowledge of them. So, all right, so now let's spend more time on the bacteria. Our knowledge is much broader here. It's definitely not full, but we have more knowledge about bacteria. And for most of us, you're going to have more involvement working with bacteria than you would with the archaea. Again, I'm not discouraging people away from the archaea. I would encourage people to go into that research. But those of you heading towards healthcare, bacteria are a big issue. Environmental folks, bacteria are a big issue. So this is why I want to spend a little more time on the bacterial diversity out there. Okay, so bacteria, I said I'm not going to ask you guys focus on this stuff here just because this is a moving target. What I want you to focus on is identifying bacteria and the key features. So let's start with shape. Basic shape. There are three general shapes: Bacillus, Coccus, Spirillum. Use those terms. Do not say, oh, I have a round-shaped bacteria. As a biologist, as somebody who is potentially heading towards healthcare, medical field, whatever, use the correct terminology. So if you've ever had an infection, the doctor may have said, oh, you have streptococcus, pneumococcus. They don't say you have pneumo-round bacteria infection. They're telling you the shape when they use these terms: Bacillus, Coccus, Spirillum. Now, Bacillus means rod, kind of a pill shape. Coccus means spherical or round. And Spirillum, kind of this squiggly or spiral. So I want you guys to use the correct terminology here. If you're describing this to a physician, you do not tell them, oh, under the microscope, I saw a round bacteria. Please don't embarrass yourselves. Tell them you saw Coccus, or you saw Bacillus, or Spirillum, because they're going to use that information plus additional information to then try to figure out, what antibiotic do I prescribe? The shape tells us general categories of bacteria. Right? So that's the first thing I want you guys to really, really lock in. Second thing is the surface structures on the cell of the bacteria. Now, they all have that cell wall we talked about. And one of the defining features for the bacteria is that they have peptidoglycan in the cell wall. So here's a cell wall. Let's make it a wall. Inside of that, here's a cell membrane. This is outside of the cell. So I'll just label, oh, it's hard to see that. That's the outside. If we go into the cell wall, what we'll see is this stuff helping make it strong and rigid, giving it protection and structural integrity. That is peptidoglycan. So it protects a cell, gives it shape, prevents it from busting. Now, how do we know if it has peptidoglycan? Stain it. You put different stains on it, and you guys will learn this skill when you take micro. That is a foundational skill of microbiology, doing what's called a gram stain. And depending upon the color, that tells you if you have a big layer of peptidoglycan or a thin layer. So gram stain is how we classify bacteria by their cell wall composition. So you take the shape and the color, and now you know general category of bacteria you're looking at. If you use the wrong antibiotic, you're actually feeding the bacteria and enabling it to grow and be worse. It's going to eat the antibiotic and make more baby bacteria. So we need to positively identify what the bacterial issue is if we're talking about a bacterial infection. So, okay, so color is important. Purple. Purple is positive. Gram positive is purple because there's a thick layer of peptidoglycan that will actually trap the stain. The cell wall itself is not purple naturally. It just grabs the purple stain and shows up that color because of this thick layer of peptidoglycan. So down here, this is the peptidoglycan layer. So nice and thick layer of peptidoglycan that will capture, it's actually crystal violet is the stain you'll be using in micro. And that gives it this purple appearance. Okay? Positive purple, thick peptidoglycan. Remember that. However, you can come up with a way to remember it. Make sure you remember it. Negative is red. So if your bacterial cell stains red, it's because it has a thin layer of peptidoglycan in it. So negative, oh, I got a negative score. I had a lot of red on my paper because I missed these questions. That's going to show up red. Okay, thin peptidoglycan is red and negative. So it holds a different stain. So lock that in, combine it. So if you're seeing this, I want you to be able to scientifically describe that bacteria right there. So you're looking at a slide from a patient and you see this. I want you to use the scientific terminology to describe that infection. Same way if you saw this. Some diseases we identify by seeing this. These two bacterial descriptions will describe the bacterial infections we have sitting here in central Illinois. And you'll know people who have gotten both of these. So describe it like you're telling a physician, this is what I'm seeing under the microscope. All right, so I would write it down, and if you want me to double check it in lab, by all means, bring it up. You can email me, put in discussion board, but I want you to be able to describe those bacterial infections right there. So, all right, so some other really, really neat things about bacteria. Some of them will form a thing called a capsule. So what our antibiotics do is we tend to develop antibiotics that target peptidoglycan layers to disrupt the cell wall. If you bust apart that cell wall, the bacteria collapses. Well, some bacteria will develop a capsule. This is another layer of protection that they put around the cell wall. So it's on the outside. So you got cell membrane, cell wall, capsule. Capsule can either be protein or polysaccharide layer. It varies from bacterial type to bacterial type. And that capsule acts as another defensive layer. So it's preventing the antibiotic from reaching, the green here, from reaching the cell wall and busting apart the peptidoglycan layer. So it's a layer defense. Again, think about how amazing these things are, and they're just single-celled creatures that have existed for billions of years. So we need to give them a little bit of respect because they're not going anywhere. They're not going away. So we got this layer that helps avoid immune systems and protection. Other bacteria may have little attachments. We talked a little bit about the flagella, which is that tail, but some of them have these things known as Fimbriae. Those are little sticky threads that help them attach to the other bacteria. It doesn't form a multicellular structure. It just gives them a way to hook on and attach to somebody else. So now you can get a bunch of these stuck together. If you guys have ever played with those little Velcro sticky balls, imagine shoving a bunch of them together. Now you have a larger ball. It's made up of all these little Velcro balls. That's what we're looking at with bacterial populations. They can all stick to each other. Some of them will also form little Pili. Sometimes I call them sex Pili. Those guys often are these little Fimbriae that extend out and allow them to attach and exchange DNA to another bacterial cell. So sometimes we know them as conjugation tubes as well. All right, so lots of neat features here. The endospore is a potential dormant stage for bacteria that if they go into this, they just simply shut down and go into isolation or dormancy and hibernation when they go into endospore component or endospore stages. So we'll talk a little bit more about bacterial features when we get into our next part of the prokaryotic lecture.